All right, we're going to start chapter 7, which is looking at cell respiration and fermentation. All right, so we know that living cells require energy, and we have to get that energy from outside sources, typically, because we don't make our own energy. So some animals can obtain energy from eating other animals. Some might feed on photosynthetic organisms such as plants and algae. So this is just showing you a picture of a puffin getting its food. So remember, energy is going to flow into the ecosystem as sunlight because it's coming from the sun, and it's going to go through a series of transformations, and it's going to leave the ecosystem as heat, because remember we talked about those laws of thermodynamics in chapter 6. So photosynthesis and cell respiration work hand in hand because they're essentially the opposites of each other. Photosynthesis is going to generate the oxygen and the organic molecules in the form of starch or carbohydrates, which are typically going to be used as fuel for cell respiration. So in cell respiration, you're going to use that carbohydrate and oxygen. In turn, you give off carbon dioxide and water, which is then recycled by plants through photosynthesis. So cells can use that chemical energy that's stored in those organic molecules to regenerate ATP, which we learned last time is the molecule which basically is going to be like our battery pack or powers all of the reactions in our cells. So again, make sure you watch these animations in the study area for chapter 7. There's one on energy flow and chemical recycling, and then there's a BioFlix animation that introduces you to cell respiration. All right, so again, chapter 7 and 8 go hand in hand because you need photosynthesis for cell respiration to occur and vice versa. So again, light energy is going to come in from the sun into the ecosystem. It's going to be converted to chemical energy during photosynthesis, and as a byproduct, they're giving off oxygen, which we need to breathe, and in turn, we need the oxygen and the organic molecules to undergo cell respiration to make ATP, which is going to power the work in our cells. As a byproduct, cell respiration gives off carbon dioxide and water, and then when the ATP is used up in our cells or other cells, it's used, gives off that heat energy, because remember, it's not 100% efficient. All right, so there are catabolic pathways. Remember, catabolic means breaking down, that essentially involve transferring electrons. And these transferring of electrons is basically central to the whole process of cell respiration. You're going to have a whole bunch of different molecules that act as electron acceptors in each step of that metabolic pathway. All right, so the breakdown of organic molecules is exergonic. So remember, that means it's going to give off energy, so it has a negative delta G, it's spontaneous. And there are two basic ways that we can make ATP through the respiration type pathway. So fermentation is a partial breakdown of sugars that occurs without oxygen. All right, so sometimes you can also see it called anaerobic respiration, but it's fermentation, and you get different products. You don't get as much ATP. Aerobic respiration means that we break down the organic molecules, but you need oxygen, and that's going to yield more ATP. Okay, so anaerobic respiration is similar to aerobic respiration, but it consumes compounds other than oxygen. So a lot of bacteria undergo anaerobic respiration, and so in our bodies, oxygen is the final electron acceptor. In other things like bacteria, maybe they use nitrites or sulfites or things like that. Okay, all right. So cell respiration, again, it can be aerobic and anaerobic processes, but usually when we say cell respiration, we're talking about aerobic respiration. The first part of cell respiration, glycolysis, can occur with or without oxygen. So that's why we say that. Typically, when we're talking about cell respiration, we're talking about the breakdown of carbohydrates or burning carbohydrates for fuel, but you can use fats and proteins. But for us, just to show you the basic and the principles, it's easier to just trace cell respiration using glucose, which is the main molecule. So make sure you know this equation because I will put this on a test. Hint, hint, hint. So glucose, which is C6H12O6, plus 6 oxygen molecules, because it has to be balanced, burns down, we break that down, give off energy, and you get 6 carbon dioxide and 6 water, and then energy in the form of ATP and heat. So make sure that you know this equation. So if I put equations on the test, I wouldn't ask you necessarily to write it out, but if I put them as a multiple choice or check the one that is the cell respiration, you should be able to identify this. All right. Again, we talked about redox maybe a little bit. I don't remember. But cell respiration occurs through something called redox reactions, which is oxidation and reduction. And essentially what this means is we're going to transfer electrons during these chemical reactions. And again, when you break down the bonds and transfer electrons, it's going to release the energy. And this released energy is ultimately used to make ATP. Okay. So when we transfer these electrons between reactants, we typically call them oxidation reduction reactions. I don't know why we call them oxidation and then we abbreviate it redox. No clue, drives me crazy. But anyway, it's a redox reaction. So it's coupled together. So in oxidation, something loses electrons, or we say it's oxidized. A lot of times when you see this in some chemical equations, it will go from no charge to a plus charge. That's how you know it lost electrons. In reduction, a substance gains electrons or is reduced, because if you add negatives to something, it becomes less positive. So sometimes you'll see an ionic compound or an ion like Na plus will go to Na plane. So one way to remember this is the words oil rig. So remember, oxidized is lost, reduced is gained when we're talking about the electrons. All right, so this is just a generalized redox reaction. So when you have a compound X with the electrons plus Y, they essentially switch. So notice here, X with the E minus plus Y. Y has no charge. It's a neutral atom, so it has the same number of protons and electrons. Okay. E becomes oxidized because it loses that electron, and you go from a negative charge to a 0. So you added a plus 1, now we're at 0. Y gained a negative 1, so you went from 0 to minus 1. So if something is oxidized, it loses electrons. If it becomes reduced, it gains electrons. All right, so this is just showing you an example of sodium and chloride when it makes that ionic bond that we talked about way back when in like chapter 2. So sodium is going to become oxidized because it loses an electron. Notice here in the first part of the equation, it's Na plus Cl. These are neutral atoms. Sodium loses an electron to become a positive charge. Chlorine becomes reduced and gets a negative charge, so it gains an electron. Okay, so the electron donor, whatever gives up the electron, is the reducing agent. The electron acceptor is the oxidizing agent. So it's the opposite of what we just saw. So if we go back here, because sodium is becoming oxidized, it's the reducing agent. Because chlorine is becoming reduced, it's called the oxidizing reagent because it's causing the sodium to become oxidized. Sodium is the reducing agent because it's causing the chlorine to be reduced. Okay, so they're the opposite. Whatever's becoming oxidized is the reducing agent. Whatever's becoming reduced is the oxidizing reagent. Some redox reactions don't transfer electrons, but they change where the electrons are being shared in the covalent bonds. So an example of this could be between methane, which is CH4, and O2. And we'll see that here in a minute. So again, make sure you watch that animation on redox reactions. All right, so Another key thing that I think I have later here in the presentation is anytime you have an organic molecule like a hydrocarbon, like a carbohydrate, lipid, okay, protein, especially carbohydrates, whenever you have an organic hydrocarbon and oxygen and it's going through a redox reaction, you always get carbon dioxide and water. So if you look here, notice this methane has CH4. It's the reducing agent because it's becoming oxidized. Oxygen is the oxidizing reagent because it comes reduced. So notice here you have 8 pairs of, 8 electrons, 4 pairs of electrons. Here you have 2 pairs. And when you come over here, you see now that it's switched. So carbon dioxide goes, the hydrogens go to the water, and the oxygens go to the carbon dioxide. All right. So, redox reactions typically move electrons from a less electronegative atom towards a more electronegative atom, which makes sense if you think about it, because the more electronegative something is, typically the larger the number of protons is, and electronegative is basically that magnetic type property to grab electrons. So again, something that's less electronegative isn't going to have as strong of a pull versus something that's more electronegative. Think of it as 2 big magnets. So the more electronegative atom is going to be like the stronger magnet, which will grab the electron. And when that does so, it's going to release energy that we can put to work. All right. During cell respiration, again, I said this earlier, fuel such as glucose or sugars is oxidized. Oxygen is reduced. We saw that earlier. So organic molecules with an abundance of hydrogen, like the carbohydrates and fats, are excellent fuels because they have a lot of those bonds with the hydrogens that can donate electrons. So hydrogen with its electron is transferred to oxygen, and when it does that, the energy is released and used for ATP synthesis. All right, so this again is just a summary. We saw this first equation before, and it's showing you that the, I just copied that and did it twice. So in this case, carbohydrates become oxidized, oxygen is reduced. Good thing is I'm not going to ask you to draw out the electrons and how this happens. Just know that. All right. In cell respiration, glucose or the other organic molecules, if we're using lipids, if we're using proteins, whatever we're using, are broken down, and this is always going to occur in a series of steps. Okay. So there's a couple different compounds that we use to help transfer these electrons. So electrons are usually first transferred to NAD+, nicotine adenosine something. I don't remember what it is. I don't spell it out. That's a coenzyme. So because it has a plus, when it gains an electron, it usually gains the electron in the form of a hydrogen. It is the oxidation. So notice down here now at the last line here, NAD is reduced to NADH. So anytime you add an H, a hydrogen, you're adding that electron there. So NADH is basically the stored energy form of NAD+. So essentially NAD is like your old cell phone or whatever it is. The NADH is the charged form of it, because now that hydrogen is that stored energy that once we break that bond, we can use it. So we have enzymes called dehydrogenases. So dehydro, meaning we're taking off a hydrogen, and ASE tells you it's an enzyme. So we have these enzymes that are going to facilitate the transfer of 2 electrons and 1 hydrogen to form this NADH, okay, plus. And when you do that, you're going to get a hydrogen ion released in the process. All right. So nicotinamide is the oxidized form. So here you can see it has a plus charge where the nitrogen is with the bonds there. You can see that we have the phosphate groups, etc. here with the bond. Okay, now hydrogens are going to come from the food, in this case carbohydrates in our apple. Dehydrogenase is going to reduce the nicotinamide, the NAD+, and oxidize it to the NADH if you go the opposite way. So when we get the 2 electrons, you're going to get 2 electrons and 2 hydrogen ions. Okay? You're going to get the 2 electrons that go in here to this reduced form. Now, if you kind of compare them, in the first one where it says oxidized, notice that you have 3 lines in the ring with the N+. Okay, so, and over in the reduced form, you only have 2 double lines and the N has no plus. That's where the 2 electrons go. 1 to break one of the double bonds and 1 to change the charge on the nitrogen to neutral. Okay, by doing that, we added a hydrogen. We broke a double bond, added the hydrogen up here in the ring, and then you have 1 H plus left. That H plus is going to be used later when we talked about chemiosmosis to form a gradient to help transfer electrons. All right, so the reduced NADH is going to pass electrons to what we call the electron transport chain, or ETC. This is the very last part of the whole factory, so we'll see that later towards the end of the chapter. Every time the electrons are passed down, they're passed to more electronegative carrier molecules through a series of these redox reactions. So you might go from NADH to, I think maybe a cytochrome C to something else. Okay, think of a slinky going down the stairs, but every time it goes down the stairs, it's going to something that's more electronegative, a different carrier. Eventually, it'll get to the very last step and transfer those electrons to oxygen. Okay, so you want it to go through a series of steps so that you kind of monitor it, because if it went straight to oxygen, it'd be like one big explosive huge reaction. But as it does that and transfers it, we can use that energy to regenerate the ATP. So oxygen in aerobic respiration is the final electron acceptor, and it's split into water. That's how we regenerate that to get the hydrogens and transfer the electrons. All right. So this is what would happen if we had an uncontrolled reaction. So you have the oxygen, you get the free energy. Okay, so hydrogen gas is highly flammable. So if you had the hydrogen and it's there with the oxygen and it gets exposed to heat or light or energy, kaboom, look up the Hindenburg. Okay, so if we did that, that would be bad. So by doing it in a series of steps down the electron transport chain, like those metabolic pathways, like the water turbines that we talked about last time, you can kind of control it, make a little bit of ATP at each step so that it's more efficient and you have better control. And then you still get the water. All right. So just to give you a rough idea, the stages of cell respiration, I'll give you a preview here. To harvest energy from glucose, we're using glucose as an example, you have 3 main stages. The first stage is glycolysis. Glycolysis, so glyco meaning glucose, lysis meaning breaking it down. So glycolysis is going to break down glucose into 2 molecules of pyruvate. Okay, there's a whole series of steps, but what I want you to know is at the end of glycolysis, you have 1 glucose, you end up with 2 molecules of pyruvate. Okay. Pyruvate is then converted to something, but it's called acetyl-CoA, but that's not listed here. Pyruvate is then oxidized and put through something called the citric acid cycle. This completes the breakdown of glucose, and this occurs in the matrix, the middle of the mitochondria. Glycolysis is in the cytoplasm or the cytosol. Citric acid cycle is actually in the mitochondria. All right. Once you break down everything so that you have all these electron carriers, like a whole bunch of different cell phones, right? Battery chargers. In the citric acid cycle, we do the oxidative phosphorylation, which is where the majority of the ATP is going to be made. That's in the inner membrane of the mitochondria, and that occurs through that series of steps that we saw back here in figure B. So you have a series of steps, the electron transport chain. Every time you do a turn or go down a step there, you're transferring electrons and making ATP. So glycolysis, citric acid cycle, oxidative phosphorylation. Those are the three main steps. Make sure you watch the overview of cell respiration in the study area there. All right, so again, here's the main keys here. So 3 stages, glycolysis. Now notice here that they do tell you that glycolysis is color-coded light blue throughout the rest of the chapter. Pyruvate oxidation and the citric acid cycle kind of go together. Old books used to call that the preparatory phase and the Krebs cycle, or the preparatory phase and the citric acid cycle. So this is like light orange, dark orange. And then the oxidative phosphorylation essentially is the electron transport chain, which occurs simultaneously with what we're going to call chemiosmosis, and that's color-coded purple. So make sure you color-code that in your notes, or however you want to do this, as you go through the rest of the chapter, so you know what's what. All right, and this is just showing you where what occurs. You should be able to just summarize this picture. And again, pyruvate oxidation, if you have a different book or you're looking up other resources, sometimes they call it the prep phase or the acetyl-CoA conversion. All right, so glycolysis occurs in the cytoplasm or the cytosol. Glucose is converted to pyruvate. We do get a little bit of ATP here, and because it's on the substrate, we call that substrate level phosphorylation. The electrons that we captured here were added to this NAD+, so through NADH. That's going to go all the way to the electron transport chain, and just wait until we get there. Pyruvate is going to diffuse into the mitochondria. It gets converted to acetyl-CoA. Okay. Acetyl-CoA is going to go through a whole series of steps in the citric acid cycle. Right now, you don't need to know all those. Again, you're going to get a little bit of ATP through that substrate level phosphorylation, because you're going to get it based on 1 or 2 substrate molecules with the enzymes. We're going to get more electrons taken to the electron transport chain. And again, it's like you have 2 different types of cell phones. We have NADH and FADH2. Okay, so these are both electron carriers. All those go into the oxidative phosphorylation, which is the electron transport chain. And now we have a whole bunch of electrons that we can use to just crank out a whole bunch of ATP. And because we're using oxygen, this is why we call it oxidative phosphorylation. And notice that the ATP little star thing is a lot bigger than the first 2. So that means we make a lot more there. Okay, so that's just an overview of what we're going to do. All right, so again, oxidative phosphorylation is probably 90% of the ATP generated. When you go through this, again, depending on the books and the levels, it'll tell you between 32 to 38. 36 is usually the average number by the time you put some ATP in and things like that. So 90% of the ATP generated occurs in that oxidative phosphorylation electron transport chain. And basically, oxidative, meaning we're using oxygen. Phosphorylation just means you're adding an inorganic phosphate to the ATP. Again, we do get a smaller amount of ATP formed in glycolysis in the citric acid cycle, but this is called substrate level because an enzyme is transferring a phosphate group from the substrate to the ATP and not through the electron transport. All right, so again, this is substrate level phosphorylation. An enzyme adds the phosphate to ADP, and then oxidative phosphorylation gives you this. So for each molecule glucose, the cell makes up to 32 molecules of ATP. And again, I have seen it anywhere from 32 to 38 depending on the book and how they put it in. So we'll go with what this book says, but I'll give you more specifics here in a minute. All right, so I'm going to stop this one here so that I can not bore you to death for too long, but that this can be a shorter video. So I'm going to upload this and then I will go into the next couple slides.